All right, so welcome. I'm thrilled to introduce today's speaker. In many ways, I think this is our most important grand round of the year in so many different ways. And first and foremost, this is the endowed Harvey Cohen Lectureship in Pediatrics. We normally celebrate this event with a dinner the night before for our speaker and their spouse, and for Harvey and for the whole community, and we really regret that now in the era of COVID that we're not able to do that. But of course, as always, we really want to celebrate Harvey as well as today's speaker. So, normally at these endowed lectureships, we have a lovely flyer that we pass out to the audience in person, and that sort of celebrates Harvey and his career. I think everyone here knows that he formerly served as chair of the Department of Pediatrics from 1993 to 2006. And so many of our wonderful senior faculty and most accomplished faculty were recruited and nurtured and developed by Harvey. Harvey's research interests include clinical trials in leukemia, mechanisms of drug resistance, determining the roles of proteins and predicting susceptibility to diagnosing and treating childhood illnesses in a variety of cancers. And of course, now he's just really the grandfather or our leader in oncology and palliative care, and so many of us have had the pleasure of working with him in that forum. So it's a real celebration of Harvey, and I'm so pleased to have this lectureship. Every year when we pick the speaker for the Cohen Endowed Lectureship, we highlight our young and our rising stars and our physician scientists, and such a special treat that today's speaker is Chuck. And so I'll just say a few things about our upcoming grand rounds and then tell you a little bit about Dr. Gawad. But Harvey told me when I first learned about Chuck and his work that Chuck was one of the very best fellows Harvey had ever worked with. So it's so fitting that we should have Chuck as our speaker today. But as always, I want to make sure people are aware of upcoming grand rounds. And February 12th is a very, very notable grand round. It's the Richard Berman lecture series, another one of our very important distinguished lectureships, but we're extraordinarily fortunate that Don Burnwick at the Institute for Healthcare Improvement is speaking on the topic of the moral determinants of health. And we really want to spread the news far and wide across our campus about this really, really important grand rounds and make sure that everybody's aware and saves the date. And then the following week, Dr. Nicole Tyson is going to speak about navigating contemporary contraceptives. As always, the text code for grand rounds is unique, and so just make a note here for CME. Next slide. Okay, the COVID-19 in Children series has really been wonderful. The important topics that they tackle every time and always very timely, and you can see that the next one, that are Thursdays at noon, and then this next one's on COVID and adolescence with a number of members of our faculty in both psychiatry and child adolescent medicine. Next slide. And want to make sure people take a special note about the Pediatrics Research Retreat. It's normally, of course, an in-person event. We've gotten quite good now at virtual research meetings. The MCHRI symposium was done incredibly well in a very interactive fashion. So we're going to build on that experience for the research retreat. What's important to know that's different this time is instead of it being a full day event on a Friday, it's going to be 3 half-day events. We're sort of splitting it up and hoping we're making it easier for more and more people to engage. So if you can visit the website, take a look at the programming and plan your calendar accordingly. Next slide. Oh, there it is. And that gives you a good sense, thank you, of how the talks are going to be distributed over Monday, Wednesday, and Thursday morning and an afternoon, 2 morning sessions and an afternoon session, really hoping to have as many people engaged as possible. And our keynote is Michelle Monge-Dizereau, who's going to be talking about her work in brain tumors. Next slide. And the Research Retreat Committee has done a beautiful job. I want to just give a special shout out to all the people here who spent a lot of time crafting the schedule as well as reviewing the abstracts, and there'll be a real emphasis on our trainees having an opportunity to present. Next slide. Okay, so with that, I'm going to introduce Charles Gawad. He's an associate professor of pediatrics, and he's the Tobey Distinguished Scholar for Pediatric Oncology here at Stanford. He's a practicing physician scientist. I mentioned that Harvey said he's one of the best fellows we ever had. He did do his fellowship here, and then he did a PhD in cancer biology. He's the Toby Distinguished Scholar for Pediatric Oncology, and he also was selected as one of the Chan Zuckerberg BioHub Scholars. As I think you're going to see both from the title and from his presentation, that Chuck works right at the interface of biotechnology, computational biology, cellular biology, and clinical medicine. And one of his most impactful accomplishments is he invented a new method, a whole genome amplification method for characterizing genetic variation across single cells within a tissue with really unparalleled sensitivity and accuracy. This says, I'm sure you'll hear, has really important applications for genomics of malignant cells, and it's really for this work that he's the recipient of an NIH Director's New Innovator Award. But every time I hear Chuck present, I learn about more and more applications, and among those are the very early detection of microbes that cause infections in immunocompromised hosts. So Chuck, welcome. So delighted to have you here and really looking forward to your presentation. Thank you.
Yeah, sorry. Thanks a lot for the introduction, Mary. Very kind introduction. It's really a great honor to present this lecture in Harvey's name. As Mary's mentioned, and many of you know, he's been a really important person for the hospital, but also for the mentoring of a number of people, including myself. And so it's really just a great honor to be able to present my work in this forum. And so, as Mary mentioned, I'll be talking about how we're using new biotechnologies and really with the purpose of applying them to try and improve the outcomes of our patients. So, just a quick disclosure. So the technology I will be discussing that I invented, with that we co-founded a company called BioScribe, and I am a co-founder, equity owner, and board member. So just to give you an overview, I'll give you a little bit of background on our lab and tell you a little bit about kind of our general philosophy and how we work. I'll talk about how we've used cell-free DNA sequencing and more recently a new IBM electronic nose to try and diagnose quickly and predict infections in oncology patients and how it can potentially be applied to other patients as well. And then I'll give you an overview of this new genome amplification method that we've invented and really developed over the last couple of years, and then talk about how we're applying that both kind of for research, but also clinically to really both understand biology, but also improve how we take care of our patients. And so kind of broadly, the general vision of the work in my lab is we try and really innovate with biotechnology to address really challenging questions, both in biomedical research and clinical medicine. And we really think about how those, questions and solving those questions or both, in the lab and in the clinic can have the greatest impact. And the two areas we're really focused on, again, is really understanding evolution at the cellular level. And as I'll talk about, we've learned a lot at the kind of bulk tissue level, but really getting within tissues, we're starting to learn a lot of new things. And then again, of course, thinking about how we can translate those things. And so primarily I take care of kids in the last few years have taken care of kids with acute lymphoblastic leukemia. But we kind of branched out because the technology has so many applications in so many different areas. So these are just examples of studies from my lab, just to get a sense of the type of work we do that I'm not going to talk about today. So we've done a lot of work with single cell transcriptomics, trying to understand the cell of origin of pediatric brain tumors. We've looked at the immune response within tumors and identified these special macrophages that can attack tumors. And then we've also looked at the kind of reprogramming process to try and create mature inner hair cells, with one focus being kids who receive cisplatinum, where if we could regrow these cells, potentially restore hearing. But those are kind of the type of studies that we do in my lab, just to give you a sense of the type of work that we do. And so I thought, especially for this audience, it's always important to frame our work in the context of a patient problem. And that's also how I kind of decide what I want to work on and how to do my work. And so kind of simply, the two questions, that we have in oncology, I think the two most important questions are, why are the treatments so toxic, and why don't we cure all children? And so, as most of most therapies are, non-specifically targeting dividing cells. And so we end up with side effects such as killing normal white blood cells, resulting in immunocompromised patients. And that, of course, puts them at risk for life-threatening infections. And so, for example, since cure rates for ALL, which is the most common pediatric cancer, have improved so much, infections are almost causing as many deaths as the actual disease itself. In addition, if the disease recurs and our initial treatment doesn't work, the tumors are usually much less responsive. And so what we found, and what I'll show you, is that these tumors are extraordinarily heterogeneous at the cellular level. And so getting rid of every last tumor actually is quite difficult, and explains why we need to use, more than 10 drugs to actually cure children with ALL. And so, again, I thought I would frame this in the context of one of my patients. And so, as a new faculty member at St. Jude, this was one of my first patients. At the time, she was a 2-year-old, initially diagnosed with ALL in 2015. She underwent standard treatment on the clinical trial at that time, but unfortunately relapsed after 2 years of therapy. But important to highlight, during her treatment, she had 8 admissions for infections, including 2 to the ICU for severe invasive fungal disease requiring intubation, and really had a horrible time before she even relapsed. And then at that time, Josh Wolf and I had actually started putting together this new study we called PredSeq, which was seeing if we could use a new cell-free DNA diagnostic test developed by a local company here named Carius, to try and predict infections in these patients and really prevent them from getting really sick. And so the Carius test, again, came from Steve Quake's lab, where I was a postdoc also, and was a friend of Ewan, who did this initial work, seeing that by simply going within the blood and sequencing, some of those reads actually corresponded to microbes when patients had infections. And so they worked in solid organ transplant patients, and they could see the reactivation of a number or infection of a number of different viruses. And so that was transferred to Carius, which then has subsequently developed a clinical test. And so Josh and I wanted to go about this a little bit differently, and not just say, can we diagnose infection, but can we predict infection before it happens? And so the way we designed this study is that we enrolled relapse patients, and every time a patient came into the hospital, which was frequently for relapse patients, we would collect a sample. And then when they developed an infection, we would take that sample and we would send it to Carius, and then we would also go back and say, can we predict that, and can we detect that same microbe in the days prior to when the patient was actually sick? And then importantly, we went to other samples where they weren't sick, and used those as our control samples to make sure we weren't just seeing things that we see in immunocompromised patients. And so I won't go into a lot of the details, but to our surprise, so not to our surprise, on the day of infection, so we call that day 0, we were 80 to 90% concordant with the blood culture results. What we were surprised about was that when we went back 1, 2, 3, even further, we were still detecting the infection in 60 to 70% of patients. And so this was really showing that these infections are starting and kind of brewing far before we actually see the patient and they show up with a fever. And so it really creates this possibility of developing tools where we can go backwards and really identify infections prior to when the patient is actually sick, and potentially intervene and prevent them from getting really sick. And so that work is ongoing, and Josh has led subsequent work looking at the change in cell-free DNA concentration after instituting antibiotic therapy, looked at fungal disease, looked at a number of different areas. but it shows a lot of promise in a number of areas for really diagnosing, but more importantly, predicting infection. In my lab, we've also subsequently developed a different version of the Carius test where we develop probes. So we take the cell-free DNA, and instead of sequencing everything, we develop some probes where we can go in and pull out mutations of the leukemia, but also microbes, and then sequence those and see if we can detect things with greater sensitivity. And this work has been done led by really Yuntao and Bali, who's a clinical fellow, as well as David, who's a medical student, continued to work with us. And so I'm not going to show a lot of details of this study, but interestingly, we do see both viruses and bacteria and scant evidence of fungus in these patients during induction. So these are the first 42 days are during induction. And we can see in the peripheral blood, and we also have bone marrow plasma samples, that there are these dynamic changes in the microbes that are invading these patients. And in some cases, important microbes. So we see quite a few herpes viruses being reactivated. We see polyoma viruses being reactivated. We see one patient who had a clear herpes virus reactivation that wasn't detected clinically. And so I think, this is just the first step of making the measurement. And now we have a tool to kind of figure out, are these things affecting toxicity? Can we intervene earlier? Are they changing, drug metabolite concentrations? So I think that's a really interesting area to pursue further. But one major shortcoming of the cell-free DNA technologies is generally, it takes 24 to 48 hours to get your results back. And so, during that time for predictive testing, that's really difficult, because even if you're testing twice a week, by the time you get the first test back, it's time to test again almost. And so we really wanted a way to be able to detect infections much faster. And so this project, which is new and we don't have tons of data for, just highlights the kind of unique community here at Stanford. And so this is Lucille Nixon Elementary School, which is just off campus here. And our daughters happen to be, and actually through carpooling, got to know Louisa, whose husband's in the physics department here and works at IBM. And so we talked and actually became friends and discussed our new lab within BMI across from the hospital. And she told us about this electronic nose she was developing at the Almaden Research Center at IBM. And so we recruited Yuntao, who's a chemical engineer in my lab, who has a unique background to contribute. But then we also had another friend of our daughters who is a graduate of Stanford and an AI expert. And so we recruited him to join the team. And then we recruited Agnes, who's a clinical fellow, and Catherine on the clinical side, to really bridge this gap from the hospital and the clinic to doing basic research, and all the way back to really kind of material science and other basic engineering going on at IBM. And so this concept of an electronic nose is interesting and not new. But the idea is that, within our noses, we have these sensors, and then those sensors are, the information from those sensors are integrated in our brains. And from that, we can learn what different smells are. And so similarly, these electronic noses have a sensor array, and those different sensors make measurements, and then you use artificial intelligence network or algorithms to identify which signals are unique to, in this case, Colombian coffee instead of Brazilian coffee. And so this has been the concept that's been around for a while, but one major limitation to the field has been the sensitivity of the sensors. And so that's what they've been working on here at the research park in Almaden, and this is Louisa's group here. But Christelle had been working on going from making these metal oxide semiconductor sensors better. And so this is a standard kind of electronic nose sensor. You can see it's about 12 microns thick. it has pores. It's not very smoothly or evenly distributed. And so you can see she's come up with a way to actually trim that down to 35 nanometers. And they've been able to tune the grain size and the porosity, and have shown that they can actually significantly increase the sensitivity of the sensors. And so there was reason to think that this electronic nose would be different than what they've been developing by other companies. And we were fortunate to develop this collaboration and have subsequently been working together and have a clinical study in place, collecting blood culture samples and also looking at viral infections in culture. And so again, we went and set up this, they call it EVA, the electronic volatile analyzer. We set up their prototype. We brought it from Almeida into our lab in BMI. And we've been doing experiments for the last 4 or 5 months, and have got the clinical study through and have started making measurements of bacteremic patients and comparing bacteremic patients to normal patients. These are, I think, 10 different sensors. But you can see here, these are the 2 IBM sensors that are new here. The other ones are known commercial sensors. And then when you look between the bacteremic patients and the non-bacteremic patients, we can start to see patterns. And the scales are actually different here, but we can see increases in resistance of the tracing in the IBM sensors. And so this really kind of shows promise that there is going to be signal there. And Vabu has been working on developing classifiers, and you can get a pretty good classifier here with the AUROC of 0.92. But there's still a lot of work to do. But I think really shows a lot of promise. First, we're testing it to be able to diagnose infection, but then we also want to go back to this question of whether or not we can predict infection. And so, our long-term goal for this is really if we could develop a predictive point of care test, so something that could run in a few minutes or not very long, where you could imagine patients coming into the clinic and having a blood sample taken as they're already doing and be checked whether or not they have the signal of an impending infection. But also in the hospital where, about 100,000 people a year die of nosocomial infections. And so instead of checking your blood glucose, we could actually test patients to see if they have any signs of infection developing. And so again, we're a long ways from this, but I always like to have kind of something to look at over the horizon. And if we can tackle all the challenges along the way, I think we'll have something that's really impactful. And so to go back to my patient and kind of continue the story, so she was our first patient enrolled on PredSeq. And I remember I was actually in clinic that day, and shortly after she was enrolled, she came in and had Rigers in the triage area. I went and saw her. She was clearly bacteremic, and she actually got admitted to the ICU. And when we went back to her study sample, she actually had the same bacteria 3 days prior, and she hadn't had any symptoms until she walked in the door of the hospital. And so really kind of highlighting how there's really the potential for big impact there. Unfortunately, her disease recurred after she had a bone marrow transplant. She had CAR T cell therapy, and the tumor evolved and was resistant to that. And we tried a number of different things. And unfortunately, her cells became resistant to everything. And she unfortunately passed away from her disease a few months later. And so the question is why? And really the biggest challenge I think in oncology is being able to address this challenge of cancer heterogeneity. And so when I was a clinical fellow about 10 years ago, Illumina sequencing was just coming online then. And I was really excited with this concept that we were going to be able to sequence tumors and really transform our understanding of tumors and change how we take care of our patients. What I quickly realized, though, is that these aren't monolithic tumors. They're actually quite heterogeneous. And so to really get at that issue, we really need to break it down into its individual parts. And the challenge there, though, as I'll discuss, is that the technology for interrogating and sequencing single cells, where you're starting with a single molecule and trying to read it out on a sequencer, were not very good. And so that led us down the path of trying to come up with a better way of doing it. And the main limitation, as I'll show you, was really the step where you're amplifying the DNA from this single cell. And this molecule turned out to be the key to overcoming this problem. But once we have a better understanding at this level, we can then kind of stitch those pieces back together, and really understand cancer biology in a much more detailed level, higher resolution level. But of course, along the way, we're learning a lot of stuff, and I'm always thinking about how we can use that information, and take it back either as a biomarker, as a prognostic indicator, as a drug target, some ways to improve the care of our patients. And so that's not to say bulk cancer sequencing hasn't provided a lot of valuable insights. So I was involved with this target study published a couple years ago, where we sequenced about 1,700 tumors through the NCI. And we did discover many new mutations present in bulk sample that were not previously known. And so the challenge has been, though, is that information really hasn't translated into improved patient outcomes. And so I think this is typical of what people have seen with clinical diagnostics with genomics, is that when you go broad-based, you find a lot of interesting things, but you don't really know what to do about it in most patients. And there are a subset that benefit, but for most patients, it doesn't really change what we do. And so, again, the question is why? And so, again, the challenge here is tumor heterogeneity. And so in this classic study a few years ago, they took biopsy samples from different locations in the same kidney tumor, and they did exome sequencing. And surprisingly, they found each site was quite different, and there were unique mutations. There were a few mutations that were shared, but most of the mutations were different between the different locations. Interestingly, all the locations had a mutation in SETD2, but they were all different mutations. And so it suggested they needed this SETD2 mutation to be able to survive, but they found different ways to do it. But I think this study really opened a lot of people's eyes to the challenge of tumor heterogeneity, and really how heterogeneous these tumors are at the cellular level. And again, this is still looking at tens of thousands of cells mixed together. So I really wanted to go down and understand if we even go within that one site to the cellular level, how heterogeneous are the tumors. And so, again, the major limitation has been for current bulk sequencing is that we're sequencing these tens of thousands typically of cells mixed together. And so even if you want to detect these rare heterogeneous populations that might be drug resistant, it's quite challenging. We can't determine if any two mutations occur in the same cell. And almost all the sequencing that's been done is at diagnosis. And so we know these tumors are evolving, as I presented in my patient, that they're changing and becoming, responding to the different treatments. And so we need to understand how the cells are evolving from diagnosis to as they go through treatment. And so, so I've been involved in the field of single cell genomics now for 7 or 8 years. And it's undergone explosive growth and a lot of changes. But there are essentially two major parts to single cell genomics. So one is efficiently manipulating and isolating cells, and microfluidics, whether they're droplet or valve-based or other, have been kind of instrumental. And then the other is amplifying either RNA in this case or DNA in this case. We've really needed to develop new tools because the amplifications had to be better when you went from tens of thousands of cells to a single cell. And so, again, I won't go into this study, but we're starting to learn a lot of interesting things using single cell RNA sequencing. And so we can take diagnostic samples, and Yakun and Lauren, and we've started working with Kara Davis's lab, have been looking at diagnostic ALL samples, but we're also looking at AML. We're also looking with Kara in the context of CAR T cell. But we can see different populations of leukemia cells and start to understand different characteristics of those cells. We can understand differences about the T cells in the leukemia versus normal T cells. And so there's a lot of information we're learning from that. And again, we'll have more to come in the coming months. But really what I've been passionate about is the genome, because we really haven't had good technology to look at the genome of single cells. And the bottleneck has really been the genome amplification step. And so the genome amplification typically results, the gold standard, which is called multiple displacement amplification, results in this kind of what we call jackpotting effect, where wherever you start amplifying first, you amplify a lot, and then the other areas you amplify a lot less. And so you lose regions, you get overrepresentation of regions. So this is actually a E. coli genome, but moving across. You see, if you went to sequence these areas that were overamplified, you'd sequence a lot, and these other areas not so much. And so that really limited what we could do with the data. And so again, existing technologies had poor uniformity or coverage. It wasn't reproducible. All this increased cost. And then we've been thinking for some time about how do we do many cells, and without going into the technical details, existing protocols were not in a good position to look at many cells in parallel. And so Veronica and I, and actually started with Kavya, who was a graduate student who rotated through my lab. But we looked at multiple displacement amplification, and just to walk you through a little bit of the technical part. How MDA works is that you get random priming. It uses a special polymerase that can copy really long stretches and can displace the strand in front of it. And those characteristics result in this, as I showed, overrepresentation of regions where you prime initially. And so the concept we came up with was, what if we just slow it down? And so we wanted to introduce these terminator molecules, which are just nucleotides that can't be added to. And the thought was that it would make shorter amplicons, and then those would be amplified less, and it would allow these other areas that are being overrepresented to catch up. And so that was the idea. However, our first experiments produced really low quality data. So the reads didn't map to the human genome, suggesting they were producing a lot of junk. The quality was low. The product was still larger than we had expected. And so we weren't really slowing down the reaction in the way we had expected. And so we kind of went back to the drawing board. And so this is a regular nucleotide. This is a terminator that has a hydroxyl group here, so you can't add another base to it. But what it does do is make a phosphodiester bond. One other feature of this polymerase we're using is it's really efficient at cutting phosphodiester bonds if the base on the end doesn't match. And so the idea I came up with was to swap this out for a thio group, and that creates a phosphorothioate bond, which can't be cut off by the polymerase. And so that actually made all the difference in the world. And so the percentage of reads mapping, went up close to 100%. The mapping quality, which is a measure of the uniqueness of where it maps in the genome, was about what we see in bulk sequencing. So 50 is a really high mapping quality. And so when we looked even in a bulk sample, so this is from 400 cells, you can see again with MDA this unevenness as you look across the genome, and much more evenness when we look with our new method PTA. And so again, just to show a cartoon of what we think that improvement had done. So we think that we were terminating the reaction, but that the polymerase was chopping off the terminator. And then we were ending up with something that was quite similar to MDA. But when we added our irreversible terminator, we ended up getting the reaction to behave kind of how we expected it to. And so when we just look at a couple measures of quality, so this is an area that's been a lot of people have tried to solve, and there are a number of different methods out here. So this is a number of commercial methods available. There are 5 or 6 more newer commercial methods that we've also compared it to, and our method is still far superior to all of those. But again, most of the reads are aligning to the human genome. which suggests it's not making junk DNA, it's actually copying the original template. And then the piece of data that I got really excited about was when you look at all these other methods, you're looking at increasing amounts of sequencing, and then you're looking at the fraction of the genome covered. And these two orange samples are bulk samples made of tens of thousands of cells. And you can see PTAR method is covering almost the same amount of the genome, starting from one single cell rather than tens of thousands of cells. And so we were quite excited with that. When we looked, this is walking across chromosome 1, and again, this is MDA with these spikes and valleys of coverage. This is the centromere, which neither method copies very well. And again, you get this overrepresentation and underrepresentation. With PTA, you can see, we have much more even coverage across the genome. And so the next question, of course, we wanted to ask is, can we detect genetic variation in those cells? And so again, these are the two bulk samples. And the question we ask is 3 or 4 million genetic variants, single nucleotide variants we're detecting in these cells, what percentage are we detecting in the single cells? And so again, you can see our method far outperformed the other methods that are out there. And so I was kind of at that point that we felt like we had something really special and really started to think about areas in which it could be applied. Another interesting feature of PTA is that it allows the mitochondrial genome to compete with the larger autosomal chromosomes. And so with the same amount of sequencing compared to MDA here, this is the QIAGEN MDA, we end up covering it about 400 times compared to a couple times with QIAGEN. And so I'll come back to how we've kind of taken advantage of that feature in a few slides. And so we've posted our manuscript on bioRxives, and be happy to share more of the technical details, but I thought that for this crowd, the application side would be more interesting. And so again, we knew we had something that was better than what was out there. And so we wanted to go back and ask these questions, how heterogeneous are tumors? And we know they're complex, but how complex? And so one of the first areas that we started was doing an experiment that people in the field commonly do, where you plate a single cell, you let it expand into two or more cells, and then you re-isolate the single cells. And so those should be genetically identical. And then we compare it to the original sample. And so if something was found in the original sample, we know it was germline present in all the cells. If we lost something that was in the original sample, it likely was a technical artifact. But then things that were unique to the single cells, if it was only in one cell, since we should find it in all of them, it's likely a false positive. But if we find it in multiple cells, it's likely a true unique mutation in these cells that's in a rare subset of these cells. And so we use that, again, not to go through all the computational parts of it, but we used that and took CD34 positive cord blood cells and used that approach to correct the sequencing and estimate the number of mutations in cord blood cells. And from that, we estimated there were about 1,000 single nucleotide changes in the time from fertilization to when a hematopoietic stem cell expands many times and then is eventually isolated from the cord blood samples that we take them from. And so we learned a lot from that in terms of benchmarking, but we really wanted to go back to our leukemia samples. And again, people have, there's been kind of this mantra out there that within leukemia, they're not very genetically interesting. There's not that many mutations. If you compare it to ovarian cancer, lung cancer, the number of mutations per megabase detected in a bulk sample are much lower. And that is true. However, we wanted to know, not at the bulk level, but within single cells within that sample, how many mutations are there per cell? And so we took what we learned from the previous study, and we used that to sequence a bunch of single cells from ALL samples and ask how many mutations are there per cell compared to the bulk sample. And so our bulk sample was in agreement, was close to what they had found in their bulk samples. But within the single cells, after correcting for the false positives, we actually found that there are about 10-fold mutations more per cell than is found in the bulk sample. And so you can imagine the level of heterogeneity when you multiply that by 10 to 100 billion cells that our patients have when they're diagnosed. And I think that starts to present or show you kind of the challenge in curing pediatric cancer, and really shows why we have to use so many drugs, and why we have to treat our patients for 3 years to be able to cure them. But I think another question everybody has is how does this heterogeneity influence treatment response? And how many resistant cell types are there? If we could identify those could serve as biomarkers of resistance to a specific drug. And so Yakun's been working on this project, but the idea is that we take a diagnostic sample, we have patients go through treatment, either induction therapy or exposed to drugs in culture, and then we look at the genotypes of the surviving clones versus the initial clones. And in this case, you can see the red clones became more abundant in the surviving sample than the original sample. And the idea is then the kind of bigger picture vision is that if we could define kind of drug sensitivities of clonotypes by doing enough samples, we could then go in at diagnosis and deeply sequence these tumors and say, if in this hypothetical example, if we find this group of mutations that activate these pathways, we know they would be resistant to this drug, but sensitive to these two drugs. Whereas this clone has this other phenotype, and if we detect both in the same sample, same patient, then we would want to look for drugs in which both clones are sensitive, or we might want to avoid a drug that we know a clone is resistant to. And so that's kind of the longer term vision. And so Yakun's been working on that, and this is still a work in progress, but over the last year, she's worked out a protocol where we can sort the cells in our lab, we can use a liquid handler to amplify the genome, and then we can make sequencing libraries and sequence them. And we do a combination of whole genome sequencing to look at copy number change, and deeper exome sequencing to look at all the coding mutations in the cells. And so we've done this for about 500 cells from 5 patients. And we're still working through the bioinformatics for the single nucleotide variant calling. But I will show a little data from the copy number profiling. And so again, this is walking across the human genome. So these are the relative copy number of the chromosomes. So for example, you can see here, this is a male because they have an X at 50%. If it was a female, it would be up at the same level as the other chromosomes. But we can see here, if we look closely, this little deletion in chromosome 4 that we see in all the cells. And then we also see this deletion in chromosome 6. And this is an unsorted bone marrow sample, and this is a sample sorted for the leukemia cells and then sequenced again in bulk. And so we had the patients go through, the patients went through 4 weeks of treatment, and then still had rare persistent cells. And so we went back in and wanted to ask what's going on with the copy number changes we saw initially in those cells. And so when we look at non-B cells, we don't see any copy number changes. There's some noise in the background. When we look at unsorted samples, so just take the whole bone marrow, again, we don't see any copy number changes. But when we sort out the single leukemia cells, we start to see some interesting patterns. And so we see some cells that look like the original cell, again, with the loss of 4 and 6. But that's actually a minority of the cells that we sequenced. And so we see another group of cells that, again, have the loss of 4, but now they have a loss of chromosome 11. Another group of cells here, this one had, again, loss of 4 and then loss of chromosome 7, chromosome 6, another loss over here. And then we had some that this looks like an ancestral clone that just has the loss at 4, but not other losses, and others that look normal. And so again, just really highlighting that the heterogeneity is really quite beyond what we could detect with bulk sequencing and what we really realized before. And really shows that, we're really kind of not finishing sequencing of understanding cancer, but I would argue really at the beginning of understanding the genetic changes that are really clinically important. And so again, the idea, bigger picture idea, is that we're going to not just find genetic changes, but find genetic changes associated with a clinical outcome or a cellular phenotype. So if you have this mutation, you're resistant to this drug, and really use that to try and direct therapy. I'm focused on ALL, but obviously many of these things are applicable to a number of different areas. But to get there, again, this is just a preview. Yuntao's using his engineering background, and we're making these hydrogel droplets where we're utilizing our technology to amplify cells within the droplets. And there are a number of technical challenges we're working through. But if we could accomplish this, we could actually be able to look at tens of thousands or even millions of cells in a single experiment quite readily. And so, I was initially focused on cancer, but then the question came up in terms of what other areas or what other challenges can we address with our technology. And so one area we looked at initially was much of what we know about what's mutagenic is using the Ames test, again, which is 50 years old now, and takes salmonella and sees if they revert to being histidine dependent, and so really using microbial genomics and really kind of a crude way of measuring mutagenicity. And so what we did is we took, again, human cord blood CD34 cells, we exposed them to the alkylating agent ENU, and we could see a dose-dependent increase in the number of mutations per cell at increasing doses compared to either vehicle control or mannitol, which is a common control used at toxic levels used in the toxicology field. And then we can look at the type of mutations we see. And so again, we see these T to C or T to A mutations that were known, but we also see a lower level of T to G, not so much C to G, and the C to T has a high background rate, but we do see a little bit of increase in that. And so then we can start to look for signatures of a mutagen. And so in this case, for the cytosine mutations, whether it's C to A, C to G, or C to T, they prefer to have a G after. And so it likely is at CPG sites, which could be methylated. And so again, but providing a signature. And then we see a strong de-enrichment for those, but we see a strong enrichment for the thymine mutations that have an A before. And so again, this wasn't known, and it's not really clear why that would be, but it's a clear signature of that mutagen. And so this provides a path to looking at mutation signatures of a lot of different mutagens and even chemotherapy. The question of how many mutations are we inducing with our treatment? This could be a way of measuring that. But again, it's with the genetic heterogeneity is not limited to cancer. And so we're all accumulating these genetic changes either through cell division or through exposure to mutagens that we accumulate over our lifetimes. And we're starting to learn more about diseases that we thought didn't have a genetic basis that actually do have a genetic basis. And so in this paper, they showed that AV malformations actually have activating KRAS mutations that are somatic. This paper suggested that there are actually somatic APP mutations in Alzheimer's patients. And we've been working for a couple of years now with Chris Walsh at Boston Children's, who's been looking in single neurons for a period of time and looking at genetic changes that accumulate in those cells and trying to understand how those contribute to disease. But one area where we have made a bit more progress is in pre-implantation genetic testing. And so when I moved here, I reached out to Barry Bear, who's in reproductive endocrinology and who's really been an innovator and pioneer in this area. And I told him we have a better way of amplifying DNA, and he said we need that. We're biopsying embryos, and we really need a better way to try and identify genetic diseases within these embryos, a more accurate way. And so that led us to this initial pilot project where we took 5 sibling embryos that had been donated to research. Barry did 2 biopsies on each embryo, and then we used the whole embryo as kind of the gold standard. We amplified the DNA using our technology, and then we looked at different classes of mutations. So chromosome gain or loss, single nucleotide variants across the genome, mitochondrial variants. In each case, as I'll show you, we're able to detect each type of genetic variation. And so this is just showing you one of the embryos. These top 2 are the biopsies, and the bottom one is the embryo. So we purposely included aneuploid embryos, but you can see this one has a gain of chromosome 8, a loss of chromosome 12, and a gain of chromosome 15. We could see all the biopsies in the embryos were showing concordant results. And then when we looked at the mitochondrial genome, again, these are all siblings, so their mitochondrial DNA all came from the same mom. And so they should be identical. But we found within one of the embryos, there was actually a coding mutation that was present in about 50% of reads in both biopsies and in the original embryo. And so it suggests in the egg, at some point, there was actually a mutation that occurred that had taken over 50% of the mitochondria, and what role that could play in subsequent disease, we don't know, but we're able to detect it with our technology. More challenging was when we got to the single nucleotide variants, and so we were able to detect about 95% of the genetic variants in each of the samples, and so we could then look across the whole genome and look for variants that either predispose or make them a carrier of disease or actually cause disease themselves. And so we found 5 genes that have been reported before and are likely to cause disease. So one is a well-known mutation, factor 5, that causes it to be resistant to activated protein C and increases risk for thrombosis. Another was in succinate dehydrogenase, which is a known cancer predisposition mutation. There was a mutation that's reported as probably causing von Willebrand's disease. There's this specific mutation that's been shown to be autosomal dominant and cause cryptorchidism. And then a number where they were carriers. And so it really showed me, the depth at which we can look at and try and understand and prevent genetic disease. And so when we look at all the carrier variants or what we think are disease-causing variants in red, and then create this heat map, we can see, 3 embryos are missing these 3, what we think are diseases. And then this embryo here is missing the other 2. And so if we had to select, and again, this is really a lot of hard work in progress, and this is just showing you proof of concept, not to say that this is how it would be done clinically, but just to show you the potential power of the technology to be able to really screen genome-wide in embryos and prevent the passing on of diseases in families. One area that I really care about with that is when we talk about cancer survivors. And so the study published by St. Jude and others have shown similar studies that children diagnosed with cancer actually have about 10% of them have a genetic predisposition. And so there are now hundreds of thousands of childhood cancer survivors, many of who have a genetic predisposition. And this could be a path to enabling them to not pass that gene on to their offspring. And so I think there's a lot of potential power there. And I will say that we have started a clinical study that is going through approval in collaboration with Noor, who's founded this company called Orchid that's really specializing in analyzing and interpreting the data. But we are planning to move forward once we get through all the approval processes of actually doing whole genome sequencing of isolated embryos, and then actually implant those embryos, and hopefully with the aim of leading to a healthy child that doesn't carry genetic disease. And so in the interest of time, I won't go through the last couple. I'll just give you kind of a high level view, but we can look in single cells after genome editing, and we can see off-target rates, so unintended consequences in single cells of genome editing. And we've also looked at single bacteria, and so isolating bacteria from rare places, or even sorting them out of our patient samples and sequencing single bacteria, and beginning to understand what's going on within the population of bacteria. And so that's still kind of early on, but technically it looks like it's much better than MDA. And so we've been collaborating with Alana Brito, but are really excited to move that project forward and potentially identify new and interesting bacteria. And I also skipped this, but just to say, we're looking at more than just DNA. So developing protocols to look at DNA and RNA in the same cell, look at DNA and methylation in the same cell. And it's showing, complementary information where we can detect mutations in resistant cells, but also transcriptomic alterations that could also be contributing to resistance. And so just to conclude, I hope I've convinced you that there's really an opportunity for predictive and ideally point of care infectious disease diagnostics using some of these new tools. I really think single cell DNA sequencing is going to transform our understanding of cancer biology. Our new method overcomes the real technical limitation of current technologies. And as I've shown, there are numerous applications, both for basic science and in the clinic, where I think it can have a big impact. And with that, I just want to thank especially my lab. And so Veronica is my spouse, and we work together, and she's done most of the PTA work initially. But just over this past year, much of this data was actually produced in the last year, and so or analyzed, and so everyone's continued to work really hard despite the really challenging circumstances. I had great collaborators at St. Jude, and as well as really excited about working with IBM and Alana's lab, and through some of the advancements in the technology through BioScribe. But most of all, I just wanted to kind of finish up by, again, thanking Harvey. I think, as I mentioned, he's been someone who's had a huge impact on the whole community and the hospital. But for me personally, he's someone that I've always really looked up to, not just as a scientist and a leader, but as someone who, really takes care of patients in a way that not only provides the best possible care, but provides, a uniquely compassionate perspective. And so it's really, again, an honor to be able to give the talk.
Thank you, Tuck. That was wonderful, and so nice to see the large number of people attending this grand round, and to see that they span a lot of our different divisions. And I too want to thank Harvey, because I know he was really critical to getting you here. So we owe him a big debt of gratitude. Okay, so it looks like we have some good questions. We'll start, actually there's a couple new ones. So one from Peng Wu, fascinating talk. As you know, IVF is associated with predisposition to BWS, which is also associated with hepatoblastoma. Does this relate to what you're finding with the clonal analysis of pre-implantation embryos?
Yeah, so we haven't looked at that specifically, and I don't think we have enough embryos right now to be able to answer a question like that. Um, I also, and I think, imprinting is also a big part of, so changes in DNA methylation are also a big part of some of these issues. And so we have a method that's working in bulk samples looking at methylation and DNA sequence variation in single cells. And one place I want to apply that is in early embryos. So I think it's still an open question.
All right, thank you. So we're a little short on time, but I am going to take two questions that are one for obvious reasons, and that is it's from Harvey, and he congratulates you on your work and wants to know if you have data to answer the question as to whether relapse is due to pre-existing clone or a genetic change that occurs during treatment. Great question.
Yeah, so jury's still out. That's one of the questions we really want to answer with this study. So I would say, I don't know. And it raises the big challenge of, how much harm are we doing with our treatment where we're inducing lots of mutations and could be creating new clones. And so I think that's a really critical question to answer, but we still don't, we need to look at a lot more patients, I think, to answer that question.
And then also timely, Michael Jang extends his congratulations as well and wants to know if you've applied anything to COVID infection prediction.
Yeah, so we did our first experiment last night, actually, where, so for biosafety reasons, we can't work with COVID. But as a proxy to that, we are working, and that was part of the initial motivation for the study. But we were working with non-SARS coronaviruses in my lab, as well as flu and metapneumovirus. And we're starting out in culture and infecting cells and then taking the fluid from that and seeing if we see a difference. If we do, then that would be kind of motivation to start looking at patient samples. But again, there are more biosafety and kind of technical limitations there, but definitely something we're interested in.
Good, thank you. And then the last question is the risk of biopsy for embryo viability and impact on successful implantation and birth to full term. A question from Ron Ariano from neonatology.
Yeah, so Barry would be better to answer that question, but this is already done clinically. I think the risk is quite low. I think the field's come a long way. The only difference we're doing, so all the upfront part is already standard of practice clinically. All we're doing different is using our technology once we have the biopsy and then getting much better data than they were getting before. But the other parts are all kind of standard of care for pre-implantation genetic testing.
No, thank you. So one of the sort of the benefits of COVID is just that, we do a better job now, not just with recording grand rounds, but really with disseminating it. So we're going to make sure this gets sent to the Cancer Institute, to maternal fetal medicine, to infectious diseases. There's a lot of different people who I know will be excited to learn more about this work. So thank you, Chuck. I'm so glad we ended with this wonderful picture of Harvey, and congratulations on all the great work.
Great. Thank you.
Thank you.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.